Welcome to a new session of Biology by Cloud Tutors, a program for 10th standard students to prepare for board exams. In today's class, we will be discussing about the chapter How do organisms reproduce? This chapter comes under biology, which carries about 26 to 27 marks in your entire science paper. The weightage for this chapter is 5 marks. Important tips: types of reproduction, modes of asexual reproduction, sexual reproduction in flowering plants, human reproductive system. These are important topics in this chapter. So drawing and labeling of diagram is important. So which diagrams can come from this chapter? Bisexual flower, fertilization in flower, germinating seed, reproduction in amoeba, hydra, yeast, rhizopus, planaria. These diagrams are important. And when we talk about human reproduction, there the human reproductive system, both the male reproductive system and the female reproductive system, can come from drawing as well as labeling of diagram. So what are we going to study today? When we talk about reproduction, there are two types. One is asexual, the other is sexual reproduction. In today's class, we will be focusing on modes of asexual reproduction, which includes binary fission, multiple fission, fragmentation, regeneration, spore formation, and vegetative propagation. Also, we will be talking about sexual reproduction in flowering plants. So there we will be discussing about structure of a typical bisexual flower, the different stages involved in sexual reproduction in flowers. That we'll be discussing in today's class. So types of reproduction. So there are two types of reproduction. One is asexual, the other is sexual reproduction. So what is the difference between sexual and asexual reproduction? Based on the number of parents, in case of asexual reproduction, there is only one parent involved. Whereas in sexual reproduction, two parents are involved. A male and a female is involved. So what do offsprings look like in case of asexual reproduction? In asexual reproduction, the offsprings will be identical to their parent. Whereas in sexual reproduction, they will be different from the parent but similar. They will only be similar to their parents but not identical in case of sexual reproduction. So what about the DNA? So the DNA will be same as the parent in case of asexual reproduction. Whereas in sexual reproduction, since there are male and female gamete involved, so what will happen here? Here the DNA is a combination of two parents. The combined DNA of two parents is seen in case of sexual reproduction. Modes of asexual reproduction. So if you see the modes of asexual reproduction, let's understand what is fission. Fission is nothing but a cell breaking down to form many cells. That is called fission. Here you can see that this cell has broken down into two cells. So what has happened? This we call it as binary fission. This is called binary fission. So this is observed in case of bacteria. So let's understand how binary fission happens in a bacteria. So we have a bacterial cell here, right? And this will be the genetic material. So that is the genetic material. So what will happen? First, the genetic material is going to divide into two parts. After the separation of the genetic material, here you can see a cell plate will be formed. After that, Once the genetic material is divided equally among the two daughter cells here, then the cytoplasmic division will take place. After the cytoplasmic division, you can see that here daughter cells are formed. Daughter cells are formed. So binary fission in case of amoeba, you can see that here is an amoeba cell. It is irregular in shape, right? So this amoeba cell is undergoing binary fission. You can see how it is broken into two cells, forming two daughter cells. So what will happen here? Here we have the parent cell. So this is the parent amoeba cell. In that what will happen? The nucleus is going to divide. Nucleus is going to divide. After that, what will happen? The cytoplasmic division will take place. After the nuclei division will be the cytoplasm dividing. Cytoplasm is going to divide, and then the daughter cells are formed. Daughter cells will be formed. In this way, through binary fission, reproduction takes place in amoeba. So let's see which are the other modes of asexual reproduction. So in other type, you can see here, in case of the paramecium, okay, you can see how the cell division is happening here, right? How the fission is taking place. So if you have to see in detail, so if you see the paramecium cell, you can see that the outer surface has many cilia, and here when you see the nucleus, there is macronucleus that is larger in size, and there is micronucleus. So this is the micronucleus, the small one here. Macronucleus and micronucleus. First what will happen? Amitotic division of the macronucleus takes place. This macronucleus is going to divide first. After that, what will happen? The micronucleus is going to divide by mitotic division. That is, equational division will happen. So by mitotic division, the micronucleus is going to divide. Meanwhile, what will happen? The oral groove will start disappearing. Once the oral groove disappears, newly formed oral grooves will appear. So these are the newly formed oral grooves. So newly formed oral grooves will be formed. After that, the contractile vacuoles will also be newly formed. So you can see contractile vacuoles for the two daughter cells here. So after that, the cytoplasmic division will happen, forming two paramecium cells. So this is how binary fission happens in case of paramecium. Next we have a multiple fission. Multiple fission means one cell is going to break into many cells. So here we are seeing multiple fission in case of plasmodium. So here we have schizosont, that is the cell here. Now this is the nucleus. So what will happen first? The nuclei is going to divide to form daughter nuclei. Nucleus is going to divide to form daughter nuclei. Further, this nucleus will start dividing into many nucleus. Now you see that this cell contains many nucleus. So once it is mature, what will happen? The schizont wall is going to rupture. And when this wall ruptures, the nucleus comes out with the portion of the cell wall. With the portion of the cell wall, the nucleus is going to come out, and that forms merozoites, means the new plasmodium cells are formed. This is how multiple fission happens in case of plasmodium. Coming to fragmentation. So fragmentation happens in Spirogyra. In Spirogyra, the propagation or reproduction is by fragmentation. So if you see the structure of Spirogyra, you can see that it is filamentous. Okay, it has a filamentous structure. So what will happen to this filamentous structure is that they break down into fragments. You can see fragment 1, fragment 2, fragment 3. Now these fragments will grow to form new cells, or new spirogyra will be formed when these fragments will start growing. So these are the new organisms formed. New spirogyra formed from the fragments, new organism is formed, and the process is called as fragmentation. Next we have regeneration. So in regeneration, here regeneration happens in case of planaria. We have taken the example of planaria. So in planaria, what will happen? Suppose there is decapitation. The head is cut over here, right? You can see the organism. So this is the full organism. Here decapitation has happened. The head portion is separated. Now what will happen? The head portion as well as the lower portion will develop into a new organism. So here if the cut is made here, what will happen? The head portion which is lost, that will develop. This is the new portion formed. Here again the new part formed is here. So two organisms are formed from the decapitated portion. From the cut portion again a new organism will be formed, and this is called as regeneration. This also is a mode of asexual reproduction. Next comes budding. So budding, here I have example is yeast cells. Yeast cells. So in yeast what will happen? Outgrowth will come, or protrusion or outgrowth will come from the parent cell. There is a parent cell. From the parent cell there is a outgrowth, and that is called as a bud. That is called as a bud. This bud will detach from the parent. When this bud will detach from the parent cell, there is a scar remaining. That is called as the bud scar. Bud scar will remain. So reproduction in yeast cell is through budding, and budding is nothing but a extra growth or a outgrowth coming from the parent cell, which will get detached eventually, and that detached cell will grow into a new organism. So this is budding. Budding can also be seen in case of hydra. So I have an example of hydra here. So what happens in hydra? In hydra, if you see, there is a bud coming up here. This is the bud. So what will happen? Once the bud is completely grown, that is going to get detached from the parent body. It will get detached from the parent body, and that bud is going to grow into a new organism. So the asexual mode of reproduction in case of hydra is also by budding, by the formation of buds. Now next is the spore formation. Here in spore formation, the parent plant is going to produce hundreds of tiny spores which can grow into a new plant. And where you can see this spore formation, we can see it in rhizopus as well as in mushrooms. So here we have a fungi. Okay, rhizopus is here. So here we have the part which helps in anchorage, that is called as rhizoid. It is similar to root. So rhizoids are here, and here we have the hyphae. So these are the parts present in the mycelium. So we have rhizoid, hyphae, and here you can see the sporangiophore. Sporangiophore is the structure which holds the sporangium, and sporangium is the structure which holds a spore, and spores are hundreds in number. So what happens when the sporangium is mature? It is going to burst and the spores will be released out. These spores are carried by air, and when it reaches suitable place, there again these fungus will start growing again. So this is called spore formation, which is a asexual mode of reproduction. So you can see how the spores are released here. So hundreds of spores will be released. So next comes the vegetative propagation. So vegetative propagation, in many plants, new plants will develop from the vegetative part. Now when I say vegetative part, it can be root, it can be stem, or it can also be a leaf. So these are the vegetative part. So in a plant there is vegetative part and reproductive part. Reproductive part are nothing but the flower and the fruit, whereas these are the vegetative part. So if you are going to propagate using these vegetative part, that is root, stem or leaf, then we call the mode of reproduction is vegetative propagation. So let's see how by vegetative propagation different plants propagate. So here I have the bryophyllum. In case of bryophyllum, if you see the leaf margin, so this is the leaf margin. So here in the leaf margin you can see that there are buds. These are called as the leaf buds, which are present in the margin of the leaf. So what will happen when these leaf buds, when these buds gets detached from the leaf, these buds will develop into new plant. So in case of bryophyllum, the leaf margin will hold leaf buds, and these buds, when they get detached from the leaf, they develop into new plants. So here in case of bryophyllum, the vegetative propagation is through the leaf buds. Now by root, which is that plant which is going to propagate by root. For example, dahlia and sweet potato. In this case, what will happen? The root, these are the roots. Okay, these are the roots. Using these roots we can propagate, or these plants can reproduce by using their roots. So if you plant the root of this plant, you can get many more plants. So the propagation is through root in case of dahlia and sweet potato. Now by stem, vegetative propagation by stem. So here potato and ginger can be propagated by using the stem. So here you can see, so potato is the stem modification and not a root modification. So by using the tuber of this potato as well as the rhizome of the ginger. So this is the ginger plant. So what will happen by using the tuber? Okay, so you can see here eyes, these are called as the eyes, the buds coming from the tuber. So using that we can propagate potato. So, and this is called as rhizome. So in case of ginger, turmeric and banana, there is stem modification called as rhizome. Using rhizome we can do vegetative propagation. We can get many more plants by using rhizome and tubers. Tuber in case of potato and rhizome in case of ginger, turmeric and banana. Next stem modification will be stolon. Okay, so chlorophytum, that is also called as spider plant. In this plant, if you see, this is the stolon, modified stem called as stolon. So stolon, using the stolon you can propagate this plant. So this stolon can develop into a new plant. So this plant is called as the spider plant. Here stolon can be used in order to propagate. Next we have runner. In case of strawberry plant, if you see here, this is called as runner. This stem modification here, this is called as runner. And using runner we can have daughter plants. From the main plant we can have the daughter plants. From the mother plant we can have daughter plants by using the runner. If you plant the runner will develop into a daughter plant. And runner can also be called as the stolons. Next we have artificial method of vegetative propagation. So artificially how we can do vegetative propagation? The first technique under this is called grafting. So for grafting we will need two plants. So one plant will be using as a root stock. The plant which bears the root, that is called as the root stock. And scion, the scion is nothing but the plant which I have brought it from, the piece of the plant which I have brought from another plant. So which bears root is called as the root stock or the stock, and scion will be the plant which plant piece which we have bought from another plant. So you are going to combine together, or you are going to fix it together by using a rubber band, or you can use a polythene tape can be used. So tightly you have to see that you tie it very tightly so that both are together. Both the stems are together, and what will happen, eventually they're going to grow together forming a single stem. So this is how grafting is done. So here what happens, this plant will have both the characters of the rootstock as well as the scion. So two characters of two plants can be seen in the same plant, in a single plant. That can be achieved by grafting technique. Next we have layering. In case of layering, for example in jasmine, so what happens here, you can see that the stem is touching to the soil here, or you can also place a stone or a weight in order to make this stem touch the soil. So wherever the branch or the stem is touching the soil, there the root starts developing. You can see the roots getting developed here. Right, now this grows into a new plant. Later you can cut this. So this is how the new plant will develop from the shoot which is held below the soil. First we have to make sure that the shoot is held below the soil. After that you start watering it. The roots will start developing from the shoot which is held below the soil. Root will develop, then a new plant will develop. So this can be seen in case of jasmine, and this method is called as a layering method. Layering method of vegetative propagation. Next we have tissue culture. For tissue culture we will need a tissue culture laboratory. So what happens in the tissue culture laboratory is that, so the first step is you're going to inoculate a part of the plant. It can be piece of leaf, stem or root can be taken, and that is put into a nutrient medium. It is placed in a nutrient medium and adequate temperature is provided. So this nutrient medium will contain nutrients as well as growth hormones, which is going to help this piece of plant to grow. So what will happen with the time, the cell starts dividing. Here you can see the cell division happening, right? So a mass of a cell is obtained, and this mass of cell is called as callus. So callus will be formed. First a mass of cell will be formed called as callus. That callus will develop into different parts of a plant. Okay, it will develop the root and the shoot eventually. So after that, once the callus has reached this stage, this callus can be transferred into the test tube containing nutrient medium, so that the sufficient space is provided for the plant to grow. So in this nutrient medium you can see that the root is formed as well as the shoot is formed. Once a required amount of growth, that is the root system and shoot system is obtained, after that this plant will be transferred into a pot. So it was in vitro first. So tissue culture will be in vitro, that is in the laboratory. Once a definite amount of growth is achieved in the plant, that plant will be transferred to in vivo. In vivo is the outside environment. First will be in vitro growth, then will be the in vivo growth. So this is how using tissue culture we can develop different types of plants. Plants with desired characters can be achieved by tissue culture technique. So this is an artificial method of vegetative propagation. Now let us understand the structure of a bisexual flower. So I have a bisexual flower here. This is hibiscus. So in hibiscus, if you see here, the structure basically involves the vegetative part and the reproductive part. When I say vegetative part, it is the stalk of the flower, which is called as pedicel, then the nectary, then comes the sepal. Sepals together is called as calyx, and the petals together is called as corolla. Calyx, corolla, pedicel, all that will be the vegetative part. Next comes the reproductive part. Reproductive parts are very important because they are involved in reproduction. So here we have pistil, which is the female reproductive part. Pistil is also called as carpel. So what does pistil contain? Pistil contains ovary, style and stigma. Pistil has three parts: ovary, style and stigma. Pistil is also called as carpel. So that is the female part. If you see inside the ovary, there are ovules. Coming to the stamen. Stamen is the male reproductive part of the flower. Stamen is also called as androecium. So if you see the stamen, basically it has a filament and anther. Inside the anther are the microsporangium, and here in between these we have the connective tissue. Connective is present. Then we have the microsporangium, which bears the pollen grains. So these are the parts of a bisexual flower. Why it is called bisexual? Because male and female reproductive parts are present in the same flower. That's why it is called bisexual flower. The female part is called as gynoecium, carpel or pistil, whereas the male part is called as stamen or androecium. Stamen contains anther and filament, where anther will bear the pollen grains, and the pistil will have three major parts, that is stigma, style and ovary. Ovary will have ovules. So these are the parts you have to remember for a structure of a bisexual flower. So drawing and labeling of a bisexual flower can be asked. It is important. Now let us understand what is pollination. How exactly reproduction happens in a flower. So sexual reproduction in plants. So once the pollen grains are matured inside the anther, those pollen grains will be released. Now these pollen grains will move towards the stigma of the flower. So the transfer of the pollen grain from the anther to the stigma, that is called as pollination. That process is known as pollination. Now this transfer of the pollen grain from the anther to the stigma, that can happen with the help of different agents. So pollination can be self-pollination or it can be cross-pollination. When I say self-pollination, there are two types of self-pollination. One is called as autogamy. Autogamy is nothing but pollination taking place within the same flower, in the same flower. Then it is called as autogamy. If the pollination is happening between different flowers of the same plant, which are in the same plant, then we call it as gynetonogamy. So that comes under self-pollination because it is in the same plant. Whereas when I say cross-pollination, it is nothing but pollination happening between the flowers of two different plants. Here the plants are different. So what will happen here? There is a need for pollinating agents. So pollinating agents can be biotic, means which are living, and they can be abiotic pollinating agents also. When I say abiotic pollinating agent, we have wind and water, which are non-living, which are abiotic. So wind and water can bring about pollination. They are pollinating agents. We have biotic pollinating agents, that is snail, insects, birds, bat and other vertebrates. Even animals, humans can also be pollinating agents. So these are all the biotic pollinating agents. Through pollinating agents, the pollination will happen. Now what has happened? The pollen grain has reached the stigma of the flower. Next what? Which is the next step? Next will be, after pollination, is fertilization. So in fertilization, what will happen? The pollen grain has reached the stigma. Now this pollen grain contains a sperm cell. This sperm cell has to reach the egg cell. So what happens here, that you have to understand. Fertilization is a very important process coming when we talk about sexual reproduction in plants. So what happens in fertilization is that the pollen grains germinate and form pollen tube. You can see here the pollen grain has germinated and this pollen tube is formed. Now this pollen tube has two nuclei here. One is the generative nuclei and other one is the tube nuclei. Because of the tube nuclei, this pollen tube has formed and that has reached the micropylar region of the embryo sac here, which is inside the ovule. So what will happen now? There are two nuclei here. So the pollen tube here gets into the stigma and reaches to the ovule present in the ovary. Now if you see the ovule here, inside the ovule in the embryo sac here, you can see that there are 8 nuclei here. So forming synergids, 2 of the nuclei forms the synergids, 1 egg cell is there, and in the center we have polar nuclei, and there are 3 antipodal cells. So now what will happen? The sperm cell will enter into the embryo sac here, and what will happen? One of the sperm nuclei is going to fuse with the egg cell, and the other one is going to fuse with the polar nuclei. So polar nuclei, there were 2, so it is 2n, plus again the sperm cell is going to fuse with that, so that becomes 3n, forming a endosperm. Endosperm will be formed. So endosperm will be formed, and that endosperm provides nutrition for the embryo. So what has happened? The other sperm cell has fused with the egg cell, forming a zygote. Now that is 2n. So that is why, since there is fusion 2 times, that's the reason it is called as double fertilization. One of the sperm nuclei is going to fuse with the egg cell, that is one fertilization, and the other fertilization is one of the sperm nuclei is going to fuse with the polar nuclei, forming a triploid fusion or a triploid tissue called as endosperm, which provides nutrition for the embryo. Since the fusion is 2 times, it is called as double fertilization. So this is a very crucial process in fertilization, that is double fertilization. So after pollination, fertilization. Now after fertilization, what will happen? Now we know that already a zygote is formed. After that, the developmental stages of the embryo is seen. So what happens here? Here the stalk of the flower will become the stalk of the fruit, called as the peduncle. So stalk of the fruit is formed. Now in certain fruits you can see the sepal, right? The sepal will not fall off. So you can see the, for example, in case of tomatoes, so the sepal of the flower will remain with the fruit. So it is remaining in the fruit here. Now the ovary, what will happen to the ovary of the flower? The ovary of the flower after fertilization will form the fruit. It forms the fruit. And what about the ovules which are present inside the ovary? The ovules which are present inside the ovary after fertilization forms the seed. Ovules will become the seed. Then coming to the stamen, stigma, style, all these are going to droop off. All these are going to fall off because their function is over. So after their function is over, they are going to dry and fall off. Only the ovary will develop into a fruit, and the ovules will develop into seeds. So this is about the germination of a seed. So here you can see that after fertilization, the embryo will develop into 2 parts called as the radicle and the plumule. A radicle will form the root system. You can see the root growing here. Radicle will form the root system, and the plumule will form the shoot system. You can see the plumule coming out. Plumule comes out, and what will happen with the days passing? So you can see the time lapse here. So 8th day, you can see the shoot growing now. The root is well developed. Now the shoot is growing. You can see the cotyledons are still there with the plant. The leaf is coming up. The number of leaves are getting added now. The shoot is growing in length as well. So this is how the seed germinates into a plant. 13th day. So this is the growth phase. You can see many more leaflets are getting added. So a completely developed plant you can see here. More and more leaf is added. If you see the leaf blade, it has become very broad now. It has broad leaves. So this is a well developed plant. Now, so let us practice few of the questions which can come from mode of asexual reproduction as well as sexual reproduction in flowering plants. So I have 1 mark questions. 1 mark questions are basically MCQs or very short answers have to be given. Such questions will come for 1 mark. 2 mark questions, you have to write short answers for that. After that, we have 3 mark question, 4 mark question and 5 mark question. All these are long answer questions. So let's discuss few of the questions which can appear in your exam. So 1 mark question, a feature of reproduction that is common to amoeba, Spirogyra and yeast. So here we have to find what is common in reproduction of amoeba, Spirogyra and yeast. So here they reproduce asexually. They are all unicellular. They produce only sexually. They are all multicellular. So here if you take amoeba is unicellular, Spirogyra is multicellular, then we have yeast which is unicellular. But so here when it comes to reproduction, in case of reproduction, amoeba reproduces by binary fission, Spirogyra by fragmentation and yeast by budding. So the mode of reproduction is different, right? By different modes they reproduce. But what is common among 3 is that all 3 are asexual reproduction. There is no male and female gamete involved. There is only one parent here, correct? So the answer for this will be they reproduce asexually, because binary fission, fragmentation and budding, all these are asexual mode of reproduction. So the answer for this question will be A. So the next answer, in Spirogyra, asexual reproduction takes place by, so breaking up of filaments into smaller bits, division of a cell into cells, division of a cell into many cells, so formation of young cells from older cells. So here first thing talks about breaking up of the filaments. So if you remember the structure of Spirogyra is filamentous, and those filaments are going to break into different filaments, and each of these broken filaments is going to develop into a new organism. So the answer here will be the fragmentation, which is nothing but breaking up of filaments into smaller bits. And these fragments or the bits they grow into new organism. Correct? So that has to be written for Spirogyra. The answer is breaking up of the filaments into smaller bits, and those bits or the fragments are going to develop into new organism. This is seen in case of Spirogyra. Next question. Factors responsible for the rapid spread of bread mold on a slice of bread are. So we have seen in old bread slices there is bread mold growing, black or grayish color mold will be growing. So we have to identify the factors which are responsible for a rapid spread of bread mold. So let's see the options. Large number of spores, moisture and nutrients in bread, presence of tubular branched hyphae, formation of round shaped sporangia. So these are the factors. Now we have to identify which factors are responsible for rapid spread of bread mold. So A says 1 and 3, B says 2 and 4, C says 1 and 2, and D is 3 and 4. So here what will happen? C, that is 1 and 2, large number of spores that will help in rapid growth of the bread mold. Also we have to remember moisture is very important for the fungus to grow. Then nutrients present in the bread. Nutrient is also very important because these fungus are saprophytic in nature. So they need the organic matter for growth. So answer here will be large number of spores as well as the moisture and the nutrients for germination of spore to mycelium. The spore has to germinate into mycelium, and that is possible in presence of moisture and the nutrient. So large number of spores should be there, and those spores have to germinate. Only then a rapid spread of the bread mold can happen. So the answer here is 1 and 2. So C is the answer, 1 and 2. Next question. So next question is today's challenge. So this is audience question. So the answer for this question I will not be giving. You have to find the answer. So the question goes like this: how many male gametes are produced by each pollen grain? Your options are 1, 2, 3, 4. So you have to tell how many male gametes are produced by each pollen grain. So what you can do, the answer for this question, you can answer by sending your answers to the link given here. This is the link which is going to direct you to the Telegram group. It will direct you to our Telegram group where you can give the answer to this question. So this is audience question for today. Next question: flowers with both androecium and gynoecium are called. So male part as well as the female reproductive part is present. Then such flowers are called as what? Bisexual flower, anther, stamen, unisexual flower. So since both androecium as well as gynoecium is present, it is called bisexual flower. Why bisexual? Because when I say androecium, it is nothing but the stamen. Stamen will have anther and filament. Anther will have the male gamete called as the pollen, and it also will have pistil. Pistil is also called carpel or gynoecium, and that will have the parts stigma, style and ovary. Inside the ovary will be the ovule which holds the egg cell, which is the female gamete. So here we have the male gamete and female gamete in the same flower. That's why it is called as bisexual flower. So the next question: the transfer of pollen from anther to stigma is called. So the transfer of the pollen from the anther to the stigma. So when I say anther, it is the male reproductive part, and stigma will be the female reproductive part. So this process of transfer of the pollen is called pollination, fertilization, adoption, diffusion. So what is it? So what is happening here? I have a flower here. So in this flower, from the anther, when the anther matures, pollen grains will be released. Those pollen grains will be carried by the pollinating agents. For example, I have taken a pollinator here, which is a honeybee. So this honeybee is going to carry the pollen from this flower to this flower to another flower here. So these pollen grains will end up at the stigma here. On the stigma it is going to end up. And this transfer of the pollen from the anther to the stigma, that process is called as pollination. And we all know there are different agents of pollination. Pollinating agent can be biotic or it can be abiotic. When I say abiotic, it is wind and water. When I say biotic, it can be an insect, it can be animals, it can be bat, snail, all that comes under the biotic pollinating agents. The fusion of a female reproductive nucleus with the male reproductive nucleus is known as, so here fusion of the male and female gamete, that is called as adoption, excretion, fertilization, then they say regeneration. So what is it? So if you see here, the process is called fertilization. What is fertilization now? During pollination, the pollen will come and the pollen grain will come and sit on the stigma here. It is attached to the stigma. So what will happen? The neck of this style will have chemicals secreted which is going to attract this pollen tube. So this will germinate. The pollen grain is going to germinate, forming a pollen tube. This pollen tube will have the sperm cell or the sperm nuclei. So that will come to the ovule, which contains the egg cell, and then one of the sperm cell is going to fuse with the egg cell, and this fusion of the male and female nuclei, that is called as fertilization. Next question. Two nuclei at the end of the pollen tube are called. What are these nuclei present at the end of the pollen tube called? So tube nucleus and generative nucleus. B, sperm and ovum. C, generative nucleus and stigma. D, tube nucleus and sperm. So what is it called? So if you see here, this is the pollen grain, correct? And it is binucleate, meaning to say there are two nucleus. And this nucleus is the tube nucleus. One nucleus is called as tube nucleus. The other one is called as generative nucleus. So what will happen here, if you see, this is the pollen grain. Okay, this is the pollen grain, and this will be the pollen tube. Pollen tube is this. This is the pollen tube. Now this pollen tube will have nucleus. One is the tube nucleus, other one is the generative nucleus. Now what will happen? The generative nucleus is going to divide, forming two sperm nuclei. Generative nuclei will divide to form two sperm nuclei. So the answer here, that is the two nuclei which are present in the pollen tube, they are nothing but the tube nucleus and the generative nucleus. The generative nucleus is later going to divide into two sperm nuclei. Into two sperm nuclei. Now, what are the component cells of an egg apparatus in an embryo sac? So the embryo sac has egg apparatus. Now what forms this egg apparatus? Egg apparatus will involve one egg cell and two synergids will be there. So this is the embryo sac, and let's understand the structure of a embryo sac now. If you see the embryo sac, we have the chalazal end and then we have the micropylar end. At the chalazal end there are three nuclei forming the antipodal cells, and here we have set of two nuclei forming the polar nuclei in the center cell, and we have at the micropylar end the egg apparatus, which constitutes egg cell and the two synergids. So one egg cell and two synergids together, it forms the egg apparatus. That is called as egg apparatus of the embryo sac. Next question. What is common in the following? What is common in the function performed by nucellus and cotyledon? For this, first you should be knowing what is the nucellus and what is cotyledon. So what you have to understand here is that if you see the picture what I have given here down, so this is the structure of a ovule. Structure of a ovule. So if you see the structure of a ovule, there is a micropylar end and the chalazal end, and this is called as the embryo sac. So this is the integument, the layer which is covering the embryo sac is called as integument, and this will be the embryo sac. Now the embryo sac is covered by a tissue called as nucellus, and this nucellus is nutritive in function. It is nutritious. So what will happen? The nourishment what is required for the embryo sac or the embryo to grow, that nourishment is provided by the nucellus. Okay, and if you see here, this is a germinating seed. The second, this is A diagram suppose, and this is B diagram. In the diagram B, we see germinating seed, and if you see the parts here, we have the seed coat, then we have the two cotyledons, then the embryo, right? Embryonic stem and root or embryo is present. So what will happen there? Endosperm is present here, right? In both the cases we have So here we have the endosperm. So what is the function of this endosperm in case of monocotyledon? Here you can see the endosperm. So in monocots, in a corn seed. So what is the function? Function of the endosperm is also nothing but to provide nutrition for the growing embryo. So the cotyledons and the nucellus both store reserve food material and does the work of providing nourishment. It provides nourishment. Nucellus is going to provide nourishment for the embryo sac, and the cotyledon is going to provide nourishment to the embryo. Here you can see the cotyledon, right? In bean seed is a dicot. There are 2 cotyledons present, and these cotyledon are going to provide nutrition for the embryo. And in monocots we have a single cotyledon here. Monocot, a single cotyledon. That cotyledon is going to provide nourishment to the growing embryo. Next we have 2-mark question here. What is a triploid tissue? How is the condition achieved in fertilized ovule? So first is, what is triploid tissue? That is nothing but we are talking about the endosperm, what is formed during the double fertilization. So what is happening here is that the endosperm is the triploid tissue in fertilized ovule. The triple fusion leading to the fusion of 1 male gamete and 2 haploid polar nuclei, that will form the triploid tissue. So here what you have to understand is, when I say triploid tissue, it will contain 2 haploid nuclei, 2 haploid polar nuclei plus plus 1 sperm nuclei. So together it will form the triploid tissue. It forms triploid tissue. Next 2-marker. How do aquatic plants undergo pollination? We are talking about aquatic plants. So they are growing in water. So what will happen? Few of the aquatic plants, they will have flowers which are growing in the air. So in this case, what will happen if the flowers are growing in the air? So the pollination is very easy. Wind will become the pollinating agent, or there can be insects which will become the pollinating agent if the flower is in the air. Then other plants, if the aquatic plant has flowers inside the water, in that case what will happen? The water, okay, the flower will be submerged in water, okay, and release their pollen grain into the water. When these pollen grains are released into water, the water is going to drift, right? The pollen grains will drift with the water, and the pollen grains will be carried to the feathery stigma of the female flower. All this will happen inside the water. So if the flower is in the air or it is growing outside the water, then what will happen? Insect or air will help in pollination. Suppose the flower is inside the water, then what will happen? The pollen grains will be carried by the water to the stigma of the female flower. So that is how pollination happens in aquatic plants. Next 2-mark question. What are the main layers of a flower? So we are talking about different whorls of a flower. When we say main layers, that is nothing but whorls of a flower. So there are 4 main whorls of a flower. Calyx. When I say calyx, it is made up of sepals. Then we have corolla. Corolla is made up of petals. Then we have androecium, which is the male part, male reproductive part, and the gynoecium, which will be the female reproductive part. So let us understand using this picture here. So what will happen? The 4 different whorls will be the petals. Then we have the calyx here, calyx, petals, or you can say corolla. Then we have anther and filament forming the stamen, which is the next whorl. Then we have pistil or gynoecium, that is the other whorl. So 1, 2, 3 and 4. So these are the 4 whorls of a flower. 3 mark question. What are the stages of post-fertilization in plants? So what happens after fertilization? Post-fertilization is after fertilization. So after fertilization, so after the double fertilization will be the post fertilization, and what are involved after that is that endosperm development, because during double fertilization a triple tissue is forming a endosperm. So that endosperm will develop now. So endosperm development will happen, embryo improvement will happen, then will be the development of ovule into seed. The ovules are going to develop into the seed, and the ovary is going to develop into a fruit. All this happens in post-fertilization. Development of seed, development of fruit, then the development of endosperm, and the embryo also will start developing. So these are the different steps which are happening after fertilization. Next 3 mark question. Explain the structure of the pollen. So what are the different parts of a pollen grain? So the structure of a pollen grain, if you have to see, so we know that pollen grains, they have a microscopic structure and they carry the male reproductive cell. They carry the male reproductive cell. If you see the structure here, they have the outer walled, they have a double walled structure in which the inner wall is known as the endospore or intine. You can see here it has a thin intine, thin wall known as intine, and outer thick wall which is known as the exine or the exospore. Exospore is composed of sporopollen. Okay, sporopollenin is what exine consists of, or exine is made up of sporopollenin. Then the function of the exospore or the exine is to protect the male genetic material during transportation. That is the transportation, when I say the pollen grains are transported from the anther to the stigma, correct? During this transportation, the exine or the exospore is protective in nature. It protects the pollen grain. The waxes and the proteins which are present on the pollen surface, they help in repelling moisture and helps in interacting with the stigma. So these are the function of the different parts of the pollen. And if you have to draw the structure of the pollen, you have to write the outer thick structure called as exine, then we have thin wall called as intine, and inside we have vegetative nucleus and the generative nucleus. Generative cell will be there. Inside the generative cell there is generative nucleus. So this structure has to be written. So this is the last question for the day. Describe the structure of the embryo sac of a mature angiosperm. Explain the role of synergids in it. So this is for 4 mark or 5 mark. So what you have to write for this is, you have to draw a beautiful diagram of the embryo sac. And in embryo sac there is a chalazal end and a micropylar end. At the chalazal end we have the antipodal cells, and in the micropylar end we have synergids, egg cell. Then we have in the center cell we will be having the polar nuclei. So these has to be drawn very clearly for the embryo sac. And how do you explain it? You have to tell that the female gametophyte has 8 nuclei, out of which 6 nuclei are surrounded by cell wall. Which are those 6 surrounded by the cell wall? We have antipodal cell, 3 antipodal cell which are surrounded by the cell wall. Then we have 2 synergid cells which are surrounded by the cell wall, as well as the egg cell also has a cell wall. So this 2 synergids and 1 egg cell together, it is known as egg apparatus. Then comes the chalazal end where there are the antipodal cell and the micropolar end where we have the synergids and the egg cell. Then in the central cell we have two polar nuclei. Now on maturity, what will happen to the embryo sac? On maturity, the embryo sac of the angiosperm, it will have 8 nuclei, 7 cells will be there. A single megaspore will give rise to embryo sac, hence that is called as monosporic embryo sac. It is called as monosporic embryo sac. Coming to the function of the synergids. What are the function of the synergid? Synergids are responsible for reproduction in angiosperms. During fertilization, pollen tube grows into one of the synergid. Pollen tube will be growing into one of the synergid, and then the sperm nuclei is going to fuse with the egg nuclei. The tube ceases growth, ruptures and releases the sperm cells. So this will be the function of the synergid during the fertilization process. So with this we have finished discussing the practice questions. Let's summarize today's class. In today's class we studied about the different types of reproduction, modes of asexual reproduction. This we have studied in the chapter How Do Organisms Reproduce. So in the next class we will be studying about reproduction in humans. See you all in the next class. Thank you.